Okay, it's recording. How great. Okay. Let people in the meeting know they're being recorded. People in the meeting, you're being recorded. Hi guys.
Hi Tyler.
How's it going?
Super good.
Great. Feel good. Yeah. That's how that's gonna go.
We need some more couple more shots. 2 more shots of caffeine to get them off.
That's right. That's right.
There's refreshments over there, cookies and donuts and cookies and orange juice. Please to eat something. Free food you're not eating?
I'm actually gonna go. No, I feel like I gotta get it going.
Do it.
Do.
Yeah, so that's what we are.
I'm pretty excited about the sugar.
No, why I didn't. Awesome. How's it going? Busy? What's that?
There's a lot going on. I imagine it works for you.
I'll probably turn that off.
Turn it off.
Because I'm gonna be over here.
Gesticulating?
Yeah, I'm a big gesticulator. Yes, indeed.
Thought it was only for the light.
Oh yeah, get the dimmer going. Cool. Hi guys. Oh yeah. Whew. Spotlight. Yeah, nice. We'll take it. So for people on the teams, I'm gonna turn my camera off for getting ready for everything. If something, I think it should work when I share the screen of the presentation, but if it doesn't, will someone let me know? That would be great.
Will do.
Okay, great, thanks.
Are we ready?
Ready, I think. Yes. It is time?
It's time. You ready? You want to sit down a little bit?
Okay, sure. Take it away.
We don't see your screen online. I'm sorry, I'm supposed to interject.
Tyler has a long 4-hour examination afterwards. I want to thank you all for attending this very special seminar. Tyler will be presenting the culmination of his years here at Ohio University as a graduate student in the Department of Biological Sciences. Just a little bit about Tyler. I barely know him. He came to us from the grand state of Michigan. He loves Michigan.
You don't want the video.
You give him the video.
Okay.
He joined us as a master's student, actually, and so we had discussed. Are we good? We discussed some ideas for a research project, and he wanted to work with lizards, and I said, well, you know, you should go to Arizona, try out this project, and he went on his own with, I guess it was your first field experience on your own project. He came back, he had a slew of data, and we started analyzing it, and he said, you know, about this master's gig, is it possible to ramp up, you know, get to a PhD? And I said, if you want to do that, we can petition for you to be accepted to the program, which he did petition, and maybe to his dismay was accepted. And for the ensuing 4 years, he's been going to Arizona collecting data on the behavior of lizards in a thermally variable environment. And I think you'll be impressed with the work that he was able to pull off while he was out there. Apart from the important work that he did in Arizona, he's also been an important contributor to our teaching mission at OU, where he's TA'd anatomy, he's TA'd intro, he has been a TA in field ecology, now he's an instructor of record in field ecology. And one of the things that you might know about Tyler is his enthusiasm, his passion for teaching, his love of bells here, and his large group of friends that support him. And so it's a great pleasure and honor for me to introduce Tyler, because this is a major passage in one's life. This is the terminal degree in biology. It's a signature of one's contribution to the field and one's potential for future scholarship, which I think you're going to do, right? Okay, we'll see. But anyway, it's been a pleasure to have him working in the lab. I've enjoyed the time we've spent together talking about research, discussing the merits of Jackie O's IPA, which are none, and our love of cocktails. So without further ado, it is my great pleasure to introduce Tyler Goerge, where he will present his dissertation topic on the thermal biology of behavior. So Tyler, take it away.
Okay, let's see here. Well, thank you, Miles, and thank you all for being here. As you said, I'm going to be talking about kind of my dissertation work, which is focused on temperature and behavior. So we'll get into it. So the world is getting warmer. Climate change is not only resulting in rising average temperatures worldwide, but also the increased frequency and severity of extreme climate events, such as heat waves and droughts. And this is exposing animals around the world to these new thermal conditions that they haven't really experienced before. And these novel thermal environments can be problematic for a wide variety of wildlife, but they can be particularly problematic for ectotherms because, unlike us, they rely on external sources to regulate their body temperatures. So these shifting thermal environments can be a problem for ectotherms because many of them are adapted to their local thermal environments. So what does that mean and why is it important? Well, body temperature in ectotherms influences pretty much every aspect of performance, from biochemical processes like metabolic rate, to whole organismal performances like activity rate and sprint speed, and even life history traits, such as growth rate. So science, for any performance that is dependent on temperature, scientists tend to use these things called thermal performance curves to characterize the relationship between temperature and performance. So for any given performance, there are temperatures below which and above which that function cannot occur. That's critical thermal minimum and maximum. The thermal tolerance breadth is between that. That's the range of temperatures at which, to some capacity, performance can occur. Then there's T-opt, thermal optimum, where performance is maximized. And so when we say that ectotherms can be are sometimes adapted to their local thermal environments, what we mean is that through either evolutionary processes or behavioral adjustments, ectotherms are able to oftentimes regulate their body temperatures pretty near that T-opt range, and so they can maximize performance and kind of do well with that. If you'll notice, the shape of these thermal performance curves tend to be asymmetrical. So they're characterized by a slow increase in performance with body temperature up to T-opt, and then a drastic reduction in performance. And this can be partially part of why climate change can be so challenging for ectotherms, because there's not a lot of wiggle room between T-opt and CT max, which is where ectotherms overheat and can die. And even if they avoid that, these small increases in temperature can result in these drastic reductions in performance that, depending on the trait, can have influences on survival and reproduction. And so it is in good interest to think about, when thinking about how temperatures changes are going to influence ectotherms, it's good to collect thermal sensitivity data on various functions. And there have been lots of studies on that, mostly on the thermal sensitivity of locomotor performance, which is either sprint speed or endurance capacity. So these are whole organismal performances that carry some ecological ramifications, such as influencing ability to escape from predators, influencing dominance. And because of these ecological applications, locomotor performance is also often used as a proxy for survival when thinking about how species are going to respond to changing environments. So scientists will go out, they'll collect sprint speed or endurance data at a variety of temperatures that gives information about CT max, thermal optimums, where are we maximizing performance, and then combined with environmental data and climate change projections, that gives us an idea of how species are going to perform these functions in a changing environment. And what we've learned from analyses like this is that tropical, subtropical, and desert species are particularly vulnerable to climate warming, because many of them are already living in environments that are near their CT max. There's less, so less wiggle room for small increases in temperature to not coincide with these drastic reductions in performance. And when that happens, when we get to that area, there's a lot of restriction in function, but also just the hours of activity that animals have available to be out and active. Okay, so we have these several studies, lots of studies that project how species will react to rising temperatures based on locomotor performance. However, the thermal sensitivity of locomotor performance might be telling an incomplete story regarding how individuals and species are going to respond to rising temperatures. So this study comes out, Gunderson and Leal, in 2015 on anolis lizards, and what they find is they map the thermal sensitivity of activity rates, which is in this black line, and then also map the thermal sensitivity of sprint speed, which is this gray line. And as you can see here, the thermal sensitivities of activity rate is not correlated with the thermal sensitivities of sprint speed, which again is a common proxy for survival. And specifically, activity rates, the thermal tolerance breadth is a lot narrower, meaning it's more sensitive to changes in temperature. And so the takeaway from this then is that we have rising temperatures may be having the potential to influence some activities or behaviors more so than sprint speed. And so when we're considering how species are going to respond to rising temperatures, collecting data on the relationships between more behaviors and temperature becomes valuable. And that idea brought me to behavior and pretty much the dissertation as a whole. So we know behavior is rooted in motor patterns that are temperature dependent. And we also know that a lot of different behaviors have these ecological ramifications. So behaviors like aggression, courtship, thermoregulation, habitat selection, mate choice, these are all behaviors that to some degree can have influences on survival and reproduction. And so I wanted to investigate the potential relationships between some of these behaviors and temperature with the idea of getting an understanding, a better understanding at least, of how species are going to navigate these changing climates from a more behavioral perspective. And so I addressed these ideas using the tree lizard, Urosauris ornatus, as a model system. And I'm going to tell you just a little bit about these lovely little guys now. So these are small-bodied desert living species that live primarily on trees and rocks. They extend over the southwestern United States and into northern Mexico. And like I said, this is a desert population, or this is a desert species. And if you remember, desert ectotherms can be particularly vulnerable to climate warming due to these, being near CT max and whatnot. The other thing about living in a desert is that there can be a lack of access to shade. So many ectotherms, including tree lizards, are thermoregulators. So what they will do is make behavioral adjustments, sometimes going between different types of microhabitats, so that they can regulate their body temperatures within a range that they prefer. And this allows them to be a little buffered from variation in the thermal environment. However, if there's lack of access to shady microhabitats, then this can become difficult for desert species. In addition, in deserts, they tend to have fewer access to resources. And so these guys are highly territorial and will compete pretty aggressively for territory and access to resources. There's also a throat color polymorphism on these guys, as you can see here. So males have these blue bellies, and females have white bellies. And males can have these blue, yellow, or orange throats, whereas females can have yellow, orange, or white throats. And there is some behavioral trait variation associated with these different morphs, but we will get into the details of that as we go along. So hold tight. Okay, so with that, we're going to get into the first chapter of the dissertation, which is how habitat use and body temperature influence push-up display rate in these tree lizards. So some lizards have a pretty unique way of communicating with one another, which are these push-up displays. So let me see if this goes. Okay, so these displays involve these push-ups that you see here. Charming, isn't it? Yeah, as well as with these push-ups, as well as head bobs, lateral compressions, and tail movements. And the lizards will use these displays to gain access to and to establish and maintain territory. And so males, especially in species that display pretty frequently. And they will display when they are engaged with other males in these kind of antagonistic encounters. They will display when engaged with females in courtship behavior. And then most often, actually, they'll tend to just broadcast, so display without direct recipients, just to kind of, let the neighborhood know, here I am, minding my space, that kind of thing. So one of the interesting things about these displays is that display rate can vary. And understanding what informs variation in display rate can give us some important information about context. So, for instance, mentioned males often will display at higher rates than females, and dominant males will tend to display at higher rates than subordinate males. The context of display can also alter display rate. So like we just said, antagonistic versus courtship versus broadcasting displays. While it is in theory beneficial to display at the highest rate possible to convey information most effectively, there are a couple potential limitations associated with maximizing display rate. So for one, these displays are very conspicuous. So in addition to these exaggerated movements of displays, a lot of times there's also some bright coloration involved with these. So like this brown anole bobbing his head, flaring his dewlap, not inconspicuous, very good for attracting the attention of conspecifics, also good for attracting the attention of predators. And that is why in several lizard species, including these brown anoles, I'm going to stop that so it stops chirping at me. They will actually decrease their rate of display when they are aware of the presence of a nearby predator. There's also potentially an energetic constraint on displays. So we know from previous work that push-up displays are related to endurance capacity in lizards, which we know also is thermally sensitive. And so because of this relationship, we could also might expect that push-up display rate would vary with temperature in a manner similar to kind of a thermal performance curve shape like we might see here. That makes sense to me, at least, but there's actually very little data collected on the relationships between temperature and display rate. In fact, only one other study that I know of in 2017, Orden Stamps, looked at the relationship between display rate and ambient temperature and found that in an Anolis species that observed display rate matched relatively the shape of a thermal performance curve. So I wanted to investigate the relationship between display rate and body temperature in our tree lizards, while also considering a few other abiotic and biotic factors. So in addition to looking at temperature, I was interested in how perch site variation might influence display rate. So perch site variation meaning what microhabitats are the lizards displaying from. Because like we just said, different microhabitats can have different levels of exposure and thermal properties associated with them that could realistically result in different levels in visibility to conspecifics and predators. And so maybe there's some display rate alteration there. I also included thermal preference, which is T-pref. And what that is the body temperature that a lizard will select in a thermal gradient given a range of choices without any costs or ecological constraints. And the reason I was interested in this here was because a lot of times T-pref tends to be co-adapted with T-opt, which is the temperature at which performance is maximized. And so maybe it is that lizards that prefer higher body temperatures are also displaying at higher rates. I also included sex because males are more territorial than females in the species, as well as dominant status. So mentioned the throat color polymorphism here. Previous work has demonstrated that these blue-throated males tend to be dominant over yellow and orange-throated males. They're more aggressive, they hold larger territories. And so I figured that could also have an influence on display rate, considering display rates are used to maintain an established territory. And then I included social context, again, courtship interactions, antagonistic interactions, or just broadcasting without a direct recipient. So for this project and for all of what I'm going to talk about today, I did my field work at the Appleton Hotel Research Ranch in Elgin, Arizona. This is an arid desert grassland, and my study site is populated by these live oak and mesquite trees, which are pretty good quality habitat for a desert. They offer access to shade, some habitat complexity, and oftentimes access to decently high quality food items, as compared to what we also have in the field site, which are these dead exposed trees that we call snags. And those are very highly exposed, not much shade, and get really, really hot as the day goes on. And so to characterize variation in the thermal environment, I use these models, these little I-button temperature loggers that go inside models that match about the size, shape, and reflectivity of an adult tree lizard. So what these do is that these record the temperatures that where they are placed would be experiencing in the absence of any thermal regulation of any kind. So what temperatures does the habitat offer? And so what I did is I placed sets of models in 5 different microhabitats that the lizards utilize. So tree trunks, the inner branches of trees, the outer branches of trees, and then on the surface of these dead exposed snags, and then on the underside of those snags as well. So to quantify display rate, I would transect, I would find a lizard. When the lizard would start displaying, I would record time elapsed and the number of displays. These lizards will display in bouts of 5 to 8 displays, then they'll stop and then continue to display. And so I would just record until they moved microhabitat, until they moved perch site. And I did that because I wanted my body temperature reading on the lizard to match what it was when it was displaying and not have it go somewhere else and have the body temperature change. I also recorded the microhabitat usage and social context, so if the lizard was engaged with a male, a female, or just broadcasting. Then in the lab, did standard measurements like body size, so snout vent length, tail length, mass, then gave individuals a unique identification for future reference. And then measured preferred body temperature. So again, the temperature that an individual will select in a gradient without any cost or constraint. These gradients spanned about 28 to 42 degrees Celsius. Lizards were in there for 90 minutes, taking body temperature readings every 10 minutes. T-pref was calculated as the average body temperature over the course of the trial. whereas T-set is the middle 50% of body temperatures selected. So to assess what factors influence display rate, I built and compared some mixed effects models with lizard ID as a random effect to account for repeat observations of display. I built these and compared these, different combinations of all the variables that we've talked about, and compared models using AICC. I calculated the thermal sensitivity of display rate by making a thermal performance curve with a general additive mixed model, because I expected the relationship between body temperature and display rate to be nonlinear. And then we anchored that curve using previous CT min and CT max data from the population. Okay, let's get into results. So this is the environmental temperature data from this study. And so kind of the takeaways here are that early in the morning hours, 7 to 8 o'clock, tree trunks are the coolest microhabitat. Nope, said that wrong, sorry. Trunks are the warmest microhabitat, that's a difference. And then as the microhabitat starts to warm up over the course of the morning, the tree trunks then become the coolest available microhabitat, whereas the exposed snags become the warmest. For reference, the horizontal lines there are the average T-pref that we recorded, as well as the T-set of the population. So you can see that even at its warmest, tree trunks don't really reach T-set, and the snags really highly exceed them. So, okay, so when we modeled what best describes display rate, this is a lot, sorry, we'll get there. When we modeled what best describes display rate, we found that an interaction between body temperature and microhabitat was the best model for that. And so what we have here is we have body temperature on the x-axis, we have display rate on the y-axis, and then we have each of the 5 microhabitats that we collected lizards from and observed from as different colors here. And the main takeaway is that lizards display at significantly different rates on 3 different microhabitats. So tree trunks, which is this purplish line, where body temperature increases and display rate decreases. On inner branches of trees, where display rate increases with body temperature. And then on these sunny snags, these exposed snags, where there is no significant relationship between body temperature and display rate. And we'll come back to that and talk a little more about that in a little bit. Thermal sensitivity of display rate. So this is the thermal performance curve that we got. So there's a couple things to note here. This is a significant relationship. So temperature does have an influence on display rate. We can also see that there is a wide range of temperatures where display rate is pretty high. So the 90% thermal tolerance breadth is the range of temperatures at which display rate can occur at 90% of maximum capacity. And in this model, and what we found is that this 90% thermal tolerance breadth spans a 16 degrees Celsius. So that's a really wide range of temperatures where lizards are able to display at a high rate. And that's something to kind of keep in mind as we talk more about kind of what drives display rate. A couple other things is that the thermal tolerance breadth did include T-pref, but T-opt, the temperature at which display rate is maximized, is below T-pref. So a few things there. So what could explain the variation in display rate that we observed? So I think that there could be a couple things going on here. So we know from our temperature models that the microhabitats have different thermal properties. And we also know that some habitats are more exposed than others, which could influence visibility of both predators and conspecifics. And based on understanding that we know that lizards can alter display rate from other studies based on the presence of predators and conspecifics, there could be some kind of interaction there going on. So if we look at the 3 microhabitats that the lizards displayed at different rates in, so this is isolated from the bigger graph. We have sunny snags, inner branches, and trunks. Here we have the relationship between body temperature and the time of day. So over the course of the day, how are those body temperatures changing? And then at relative activity rates of each microhabitat. Solid lines are significant relationships. Dotted lines are insignificant relationships. So in sunny snags, we had by far the highest activity of all the microhabitats. As in the early mornings when it's cold out, lizards are using these really highly exposed sites to bask and warm up with. So here, body temperatures increase throughout the day, but display rate stays consistent throughout the day. And the other thing to note is that if we take a look at sunny snags compared to the other microhabitats, they stay consistent through the day and they stay relatively low throughout the day. And I think that it's as by far and away the most exposed microhabitat of the bunch, with really no access to any refuge besides the odd crack in a bark somewhere. These are really highly exposed predators, especially visual hunting predators of the tree lizards. And so I think that there might be some alteration of, keeping display rates relatively low in this highly exposed microhabitat. On tree trunks on the bottom there, we have display rate decreasing with body temperature. So tree trunks also have a relatively high rate of occupancy during the early morning hours, but then as the day goes on, occupancy starts to drop off. So display rate could be highest at temperatures on trunks, simply because this is when lizards are there the most, giving an increased number of potential recipients. The other thing to note is that trunks in these early cold hours the display rate is really quite high compared to any of the other microhabitats. And so trunks are highly exposed as well when they're on the trunks, but they also offer a lot more access to refuge. So you can go into the tree bark, you can go up into the branches, which are really kind of dense. And so we have an exposed habitat that's also much more safe from predators in terms of available refuge spots. So there could be some, maximizing of display rate there for that reason. And then on inner branches, we have an increase in display rate with temperature. So as the morning gets late and it starts to get hot, lizards will start to take refuge in these shady microhabitats as compared to the more exposed snags and trunks. And so similarly to trunks, I think that this relationship with display rate and body temperature might be low at low temperatures, just because lizards aren't occupying these habitats at those times of day. There's not a lot of recipients. So then as lizards again shift perch site to inner branches as the day goes on, display rate increases with body temperature. So we have evidence of display rate being influenced by this interaction between temperature and microhabitat, which I think is at least partially being driven and explained by the shifting social structure of conspecifics over the course of the day. We don't have any evidence of sex, throat color, social context, body size, or thermal preference on display rate here. Okay, so we are going to move on to chapter 2, which I kind of got to by thinking about, body temperature, thermoregulation, and social structure. And so what we're going to be talking about now is the influence of conspecifics on thermoregulatory behavior in these tree lizards. So we already talked about thermal preference. Again, it's the body temperature that lizards select when given a free choice in the absence of any cost or constraint. And these are useful for several applications. So like we said earlier, they're often adapted with optimal performance, so maximizing performance at these temperatures. They can also be used to measure how accurate or effective individuals are at thermoregulating compared with habitat data. And they can also be used in projecting local extinction risk. So when temperatures start to rise beyond these range of T-pref values that lizards are selecting, we assume that available hours of activity are going to drop. So these applications are useful, but like we said, T-pref is measured in the laboratory, and under this kind of ideal scenario where there are no ecological costs or constraints, and that never happens in the field at all, because there's always some kind of cost or constraint associated with thermoregulating. So time spent thermoregulating is time that can't be spent doing other things like foraging, territory defense, or mating, and also basking associated with thermoregulation can increase predation pressure. And so these costs and constraints are often considered when we're modeling optimal thermoregulation strategies and behaviors, but they're not actually ever really considered when collecting T-pref data. And so introducing realistic constraints to collection of T-pref data might provide a more realistic representation of how these animals are thermoregulating in the field. And so I introduced a constraint to thermoregulation in the tree lizard, and I selected the presence of conspecifics. So conspecifics, just other lizards. So this is applicable because these lizards are densely populated with sometimes multiple males and females sharing the same tree. It's not very big either. And so because thermal quality is a large driver of habitat selection, there are likely some high levels of competition for kind of these ideal basking sites. So I was interested specifically in how sex and throat color would influence thermoregulatory interactions. So remember, we talked about how blue males are dominant or tend to be dominant over yellow and orange males, and there's a good amount of data and studies kind of talking about the dynamics between those three morphs. However, there are also a lot of there's a lot of geographical variation in what morphs are around. And in my population, we have a lot of these guys, these yellow and blue male morphs. And there's not as much data on kind of the role that they play ecologically. What we do know is that they tend to be dominant over yellow males in thermoregulatory interactions, or sorry, in dominance interactions. And likewise, these orange-blue males, which we also get, tend to be dominant over these guys. And so based on this idea that the amount of blue in a throat influences dominance status, I categorize my male groups into 3 categories. So blue males, these males with some blue in the throat, so these orange blue and yellow blue, and then the males that have no blue in the throat at all, into those 3 categories. And so I measured T-pref in the same gradient, but for every lizard that I measured, I also staged these paired contests, with one being with a member of the opposite sex, one being with a member of the same sex and same color morph group, and one being a member of the same sex and different morph, again, using those male groupings that we talked about. So I randomized these trials, the order, and separated them by a day. And then to analyze how conspecifics influence T-pref, I used mixed effects models, including body size as covariate. So I looked at tau. Oh, we got someone. Okay. I looked at these thermoregulatory interactions for males and females, and then also broke them down into morph, which we'll see in a minute. So going into this, I kind of thought that males conspecifics would engage in high-level territory, resulting in displacement from optimal positions in the gradient. I also thought that male color morph would influence the magnitude of displacement, with again these yellow and orange males maybe being displaced the most and these blue males being displaced the least. I also thought that female conspecifics would compete for basking territory, but less so than males because they're less territorial. And then finally, that when males and females were in the gradient together, that maybe males would prioritize courtship behaviors over these territoriality behaviors and maybe follow females around in the gradient. That's what I thought. We'll see if it happened. So what did we find? So these are the results for males and females, males in the blue line, females in the red line. And what we have here is T-pref on the y-axis, solo T-pref values here, when paired with the opposite sex, when paired with the same sex, same morph, and same sex, different morph. And what we find in, let's do males, is that so when males are in the gradient by themselves, they have significantly higher preferred body temperatures than when they are in the gradient with males of the same morph and males of the different morph. And females in the gradient by themselves have significantly higher body temperatures than when paired with males or with females specifically of the same color morph as they are. So we'll take the female line first, and we'll break up that red females line into the distinct morphs of the females. And what we get here is one significant result of in orange females, which is this middle orange line, solo T-pref being significantly higher than when they are in the gradient with males. So we have orange females being significantly displaced from their optimal thermoregulation position by males. And if we want to kind of take a look at a little further what male morphs specifically are kind of influencing that the most, we can break this up into female solo. This is female solo temperatures, and this is orange female temperatures when they're in the gradient with blue, blue yellow, blue orange, and yellow orange males. And what we find there is that this decrease in thermal preference is being driven by when orange females are in the gradient with these yellow and orange males. Okay, so now we're going to look at males. So if we break up that blue line, and again, overall males are significantly decreasing T-pref values when they're in the gradient with other males. This is our morph grouping. So we have blue, yellow, and blue orange males in the green. We have blue males here, and we have yellow and orange males here. So what we see is blue, yellow, and blue orange males consistently having the highest body temperatures, and yellow and orange having the lowest, although there's kind of a similar patterns in all of the morph groups. So in blue, yellow, and blue orange males, the green line, they are significantly decreasing their T-pref values when they're in the gradient with males of also blue, yellow, and blue orange morphs. And when that is the case, the smaller of those two males is being displaced the most, 8 out of 10 times in those interactions. And then when they're back in with different morphs, the T-pref values go back up. So in I think, yeah, so in yellow and orange males, which is the orange line, T-pref is significantly higher in the solo than when they're with males of different color morphs. So they're being significantly displaced when in the gradient with males of blue, yellow, or blue orange throats. So I know that was a lot, sorry. As we expected, we'll recap here. So as we expected, males were significantly displaced from their solo T-pref values when they were in the gradient with other males. And there was an influence of color morph. So as we expected, these yellow and orange males were displaced the most and had the lowest body temperatures. Kind of somewhat surprisingly, the blue, yellow, and blue, orange males were kind of dominant over the blue males in these interactions. And so again, the only time they were significantly displaced is when they were with males of the same color. And at that point, body size kind of seemed to serve as a tiebreaker. But then again, when they're integrating with any different male morph, their T-pref shoots back up closer to their solo T-pref values. When males and females shared a gradient, we did not see males following females around like we thought. So females never significantly displaced male T-pref values, while the females, and especially the orange females, were displaced when they were in the gradient with males. So rather than courtship, it seems like males were just kind of engaging in territoriality behavior with females. This could be because many females were gravid at the time of running this study, and so maybe females were not as interested in courtship. Alternatively, because females were gravid, they were not really interested in courtship either, and they just were avoiding males at high levels, which could be very likely. So we'd have to run that again in a less reproductive season to kind of see what's going on there. And finally, females did displace each other significantly when they were in the gradient, and specifically, females significantly displaced each other when they were in the gradient with the same morph. In other female polymorphic lizards, throat color tends to correlate to reproductive traits and reproductive strategies. And a lot of times, sometimes female morphs will actually space themselves out so that they are decreasing contact as much as possible with females of the same color morph, so there's not as much spatial overlap with someone that shares the same reproductive tactics as you. And in those couple studies, they also found that when you manipulatedly, that's not a word, when you moved a female of the same morph closer to one of the same morph, they actually, their stress levels rose significantly. So there could be something similar going on here, but we just really don't know much about female Urosaurus morphs and what kind of roles they play. So overall, this gives us an idea of how the interplay between social structure and thermoregulatory behavior could influence the body temperatures that lizards are actually experiencing in the field in these densely populated populations. Okay, so thinking about, the effects of color morph on thermoregulatory behavior got me thinking about behavioral variation and kind of ecological differentiation between these different morphs. So that's what we're going to talk about now. So we've already touched on color polymorphism in the tree lizards, but it exists in a lot of other lizards as well as just other taxa as well. And there's oftentimes distinct variation in morphology, physiology, or in behavior between these different color morphs. And oftentimes, these distinct morphs are able to all persist in the same population due to this variation allowing situational advantages for morphs and situational disadvantages for some morphs, depending on the context. And so these situational advantages can arise through multiple mechanisms, but one of them is frequency-dependent selection that arises via alternative mating tactics. So, wow, what is that? Well, let's use an example to find out. So, this is the side-blotched lizard. There are 3 male morphs. The orange males are hyper-aggressive, and they defend large territories. The blue males are less aggressive and still defend territory, but smaller territories. And these yellow males are not territorial, and they mate with sneaker tactics, which is they will infiltrate the territory of another male, mate, and get out. These blue, yellow, and orange males are all able to persist in a population due to what's called negative frequency-dependent selection, which kind of works like a rock, paper, scissors game. So, for an example, let's say there are a lot of orange males in the population. These orange males compete really effectively against these blue males because they're more aggressive than them and they're more territorial than them, and they basically outcompete them for territory. However, their territories are so large that they are more susceptible to being sneaker mated in their territory by these yellow males. So these yellow males do well against the orange males, and if they have enjoyed greater reproductive success and there becomes more males in the population over the next few years, then that happens. When that happens, we know that they don't do well, or that they do well against the orange males, but they don't do well against the blue males because they defend smaller territories and are able to kind of keep a better watch out for these yellow sneaker males. So there becomes more blue males in the population, orange males do well, and it cycles on. So that's kind of how that works. In lesser known systems than this one, it's important to kind of quantify trait variation between these distinct lizard morphs to get a better understanding of how they diverge ecologically, kind of like this. So dominance variation in lizard morphs exists, and it often is attributed to variation in behaviors like boldness, exploration, and aggression. So for example, in the Aegean wall lizard, orange morphs are much less bold than yellow and white morphs, as demonstrated by predator avoidance behavior. And they're also less dominant than them, and they do not perform well when they are paired up with them in kind of interactions, the staged interactions. So likewise, boldness can not only influence dominance, but it can also influence ecological variables like habitat use, predation, and reproduction. So in some lizard species, bolder individuals tend to have larger home ranges, higher quality territory, have higher reproductive success, but also sometimes suffer higher predation rates because they are bolder. Likewise, exploration can influence some things, so dispersal capacity as well as survival that is kind of facilitated through rapid or non-rapid habituation to nearby predators. So despite the influence that some of these behaviors have on survival and reproduction, we don't know much about behavior strategy variation in different male lizard morphs outside of, again, a few model systems that are documented pretty well. Likewise, we know very little about thermal preference variation in lizard color morphs, even though if we're assuming polymorphic species have these different behavioral strategies, they might they might diverge in ecological, niche usage that could correspond to dissimilar habitat usage, and using different habitats might mean that we're encountering different temperatures in our microhabitats. So that we could expect some thermal preference variation between morphs that are using different microhabitats, but we really don't know much besides these I think these are the only two studies that have really looked at thermal preference variation between color morphs. And so investigating this as well as covariation between behavior or with behavior could give us an insight into kind of like morph niche specialization and differentiation. So I assessed for boldness and exploratory behavior, as well as thermal preference variation in Urosaurus male morphs. So like I touched on, I use these behaviors because they correlate with dominance and can influence things like territory defense. I categorized males into two groups based on what we talked about earlier, dominant versus subordinate. Dominant having blue throats or partially blue throats, and subordinate having no blue in their throats at all. So I expected dominant males to be bolder, more exploratory, and to have thermal preference covariation with the assumption that they're utilizing slightly different microhabitats. So I captured lizards and did the usual measurements. For T-pref this time, I included T-pref coefficient of variation, giving us an idea of the range of temperatures that lizards are selecting for. And I also included a shuttling variable, which is basically every time we took the lizard's body temperature, it changed to some degree. What I did is I added up that degree of change over the course of the whole trial to get a shuttling variable to try to quantify behavioral variation in the gradients, where lizards with high shuttling variable or values are kind of going back and forth between different temperatures, and lizards with small shuttling values are kind of picking a spot and staying there. Okay, so this is how I measured exploratory behavior in a gradient like this. And I quantified exploration as the amount of transitions that a lizard made in the 30 minutes that they had to just explore the arena. So I'll scroll through this video to show you a not exploratory lizard. Some of them will just sit there the whole time. And then other ones are, happy to move around. So the transitions range from 500 to 0. So there's that. So I measured boldness behavior in an enclosure that had a refuge box at one end and a heated perch at the other. So I allowed lizards to acclimate to the enclosure and initiate perching, at which point I simulated a predator by scaring them into the refuge box. Then I quantified, I quantified boldness as the amount of time that lizards spent both basking in the general area as well as perching on the perch for the remainder of the 45-minute trial. So I'm going to kind of skim through here because I'm short on time, but you see a lizard approaching, initiate basking, and then initiate perching, where they would tend to stay. So I calculated the repeatability of each trait by releasing the lizards and then capturing them again at least a week later and remeasuring the traits. And I allowed this 1 week time period between repeated measures to avoid confounding effects of acclimation to the laboratory. So I assessed behavioral and thermal trait variation between the status groups, and I also assessed for patterns of behavioral and thermal trait covariation in relation to a morph using a principal components analysis, which again, I think I'm a little short on time, so I'm going to talk more about that when we get there. Okay, so how do these traits differ in males? So we had 37 dominant males and 25 subordinate males, and what we find is that every single trait differed between these groups. So dominant males are more bold, they spend more time basking and perching, they're more exploratory, they prefer higher temperatures, and they prefer lower ranges of temperatures than the subordinate males, and the subordinate males are also exhibiting more shuttling behavior in the gradient. In terms of repeatability, the boldness and exploration were repeatable, T-pref-CV was repeatable, T-pref was close to repeatable except for a couple outliers that were real tricky, and then shuttling not so much. So this is our principal components analysis, which again shows the variation, covariation between all these traits that we measured and how the morphs fit in this space. And so just to kind of simplify it, basically we have 3 axes that explain all of the variation. So if we look at axis 1, which is just your traditional x-axis, what it's describing is individuals that are perching and basking, so they're bolder, are also preferring smaller ranges of T-pref variables, and they shuttle less. If you kind of see, you can see that blue males are grouped kind of this way on this axis, which is defined by lower shuttling and lower T-pref variation and higher boldness, whereas these subordinate males are more on this end of the axis. The y-axis, principal component 2, describes individuals with higher T-pref values and lower T-pref values and higher T-pref-CV and shuttling values. And then principal component 3, which you can't see here because you only see 2 of them, describes larger lizards as being bolder and more exploratory. And when we run an analysis to see if these morph groups differ in this trait space, we find that they do, and specifically they differ along principal component 1. So we have these significant differences between the groups in this covariation of boldness and thermal preference traits. We'll move on from that now. So we know that color polymorphic species oftentimes will occupy different environmental niches. So in lizards, in these tree lizards specifically, there is some evidence that dominant males utilize higher quality habitat than subordinate males. And when we looked at this study, sure enough, what we found is that 56% of our dominant males were captured in these live trees, which we consider higher quality because they have access to shade, they have more prey items and higher quality prey items. Whereas 61% of the subordinate males are captured on these snags, which again, not much exposure, not much food. A lot of exposure, not much food, and less habitat complexity. And so I think here that we have these dominant males that are selecting for higher quality habitat, and I think they could also be excluding the subordinate males from these high quality basking sites as evidenced by this covariation in thermal preference. So these guys have not only higher thermal preference values, but the subordinate males are probably going to be, as a result of behavioral differences, being excluded from these high quality sites and having to shuttle back and forth between lower quality habitats to maintain their preferred body temperatures. Okay. And finally, we are going to look, and we'll do this quickly, at how boldness behavior might be influenced by rising temperatures. So phenotypic plasticity can play a key role in buffering species from climate change. So phenotypic plasticity refers to phenotypic variation in the absence of any genetic changes. And one relevant example of this is plasticity in heat tolerance. So some species can temporarily increase their CT max during periods of heat stress in a process called heat hardening. And so what this is as body temperatures start to approach here, this thermal tolerance maximum, we have an upregulation in heat shock proteins and an increase in thermal tolerance, which in Urosaurus can be by over 1 degree. And this can provide an effective kind of short-term first line of defense when they're exposed to these extreme climate events that could be challenging and threaten survivorship. However, stress responses tend to be energetically costly, and heat hardening is no exception to this. And so a recent study showed by Anthony and Miles showed that in these tree lizards, a heat hardening response is accompanied by a reduction in locomotor performance and a reduction in thermal preference, both of which are not ideal in a warming climate. And so what we have is an adaptive, potentially adaptive response in heat hardening that also is accompanied by potentially maladaptive phenotypic shifts. And as ectotherms potentially rely on heat hardening more as climate change continues, understanding the full nature of the phenotypic relationships and shifts associated with heat hardening will help us understand its full potential as an adaptive response to climate change. And so based on this and based on some preliminary data of mine that showed that boldness behavior might be thermally sensitive, I investigated the influence of heat hardening on boldness behavior. I did the same thing as before, this time using latency to emerge as my proxy for boldness. So that was out. So this time, lower values are going to be bolder lizards because they're out of the shelter faster. And that, okay. And so then after doing this first boldness measurement, I induced a heat hardening response by warming lizards to their CT max. And then after they were there, no one died. So you put them in water afterwards, they cool right down, it's okay. But 6 hours after that, which is when the max heat hardening response occurs, I did another boldness test. And then assessed if this heat hardening response influenced boldness using mixed effects models. So I had 37 males and 31 females for this one with this distribution of morphs. And the results you see here are for all the lizards together. So what we have is the latency to emerge from the shelter on the y-axis, and then heat hardening boldness here and baseline boldness here. And so what we have is a significant increase in boldness behavior during the heat hardening response because, again, lower latency values means that they're emerging from the shelter faster. When we look at the same thing, but just in males, we find the same result. So in males, boldness behavior is increasing during a heat hardening response, whereas in females, while the trend is kind of there, it wasn't statistically significant. So what these are the average morphs for baseline and peak heat hardening. And what we see is that for the 4 males that we had to test on, they're all showing this trend of an increase in boldness behavior accompanying heat hardening and a significant increase in these yellow and blue males, which we had 20 of. And while the same idea is present in these orange and white females, I don't know what's going on with the yellow females, none of these are statistically significant. So what we have is heat hardening response increasing boldness behavior in males, but not in females. So remember, previous work has shown that a heat hardening response in these lizards is accompanied by a decrease in thermal preference, which, coupled with limited availability of shade in a desert, is going to increase competition for an already limited microhabitat. Because bolder individuals tend to have higher success maintaining and defending territory, I think that this increase in boldness behavior is likely a reflection of the increased, decreased thermal preference resulting in increased conspecific competition for these limited suitable microhabitats. This could also help explain why males increased in boldness, but females did not, because males are much more territorial than the females are. And also why we have a significant response in yellow and blue males, because we know they're a dominant territorial morph that, like I said, defends territory. So it's hard to say what the exact influence on survivorship this response could carry, but we do know that bolder individuals in general tend to engage in riskier behavior, which can increase predation risk and often does in populations where that's looked at. And so coupled with reduced locomotor capacity associated with this heat hardening response, this increase in boldness could definitely potentially increase mortality events, and it could have potential to limit the adaptive response of heat hardening as a way to increase short-term survivorship in periods of heat stress. Okay, that's what I have. I have a lot of people to thank. Miles for everything. All of my committee members, thank you so much. BIOS graduate students who have made this adventure so enjoyable. Past and present, the ones here, the ones listening in, the ones that have left long ago, all mean so much to me. So that's been great. The folks at the Appleton Hotel Research Ranch, Christina, Suze, and Ben, for letting me live with them for the summers a lot of times and making it so much fun. Funding sources, and all of you guys for being here, especially my Michigan traveling family, and Annika for skipping work. Okay. We're on time. 4 minutes for questions.
Did you see any body size differences in work?
Didn't actually, and expected to, especially in male, and male variation. Yeah, but no, didn't. Part of it could. I mean, these guys only live for like 2 years, and so it's like they have like a short adult period. There's not a lot of time to have these differences in growth rate really accumulate, probably. But yeah, some also, I should say that some populations do see them, but not in ours. So there's just a lot of variation in these things. So no, yeah.
Do you know why? Because you showed a couple of different lizards, growth or variation and stuff. Do you know like why certain hues might I don't know the dominant more composition? Is it
Like why specific colors?
Yeah, because it seems like it just really intrigues me.
Yep. And it even is that like in your source, the orange morphs are the least dominant in these interactions, and the wall lizards, they also tend to be the least dominant, but then in the side blotch, they're the most dominant. I don't know what certain colors are associated with certain behavioral strategies. Yeah, I don't know at all. Do you? No, not you. I guess we could talk about it after too. But.
Well, it just depends on which color is associated with high levels of testosterone. So
there you have it. And I guess that varies by species. Yeah, Riley.
My question actually would down off that, and it is: has anyone ever investigated if those colors are giving off some kind of UV response, like in birds?
Yep, there is some. That's starting to become a thing. So. Tends to be, especially so these guys, there is some UV associated with the different colors. I think that they all have some degree of UV, so it's not like some are giving off UV and some aren't, and that's kind of leading to, variation in that. But there are a guy that I was out there with, like last time I was out there was doing a study on thermal preference variation with considering the effects of UV and throat colors. So that's something that people are starting to look at a little more, but not me. Yeah, Brian.
What was your acclimation procedure like for just getting the lizards in the lab, and so how much of their behaviors would be reflective of body condition and levels of hunger that they've kind of
Yep. So what I did is because food availability can influence behaviors in certain things. So what I did is I would bring them to the lab, offer them water right away and food right away, give them 24 hours to kind of acclimate to the lab, have a mealworm, and then from there start the trials. And then what I try to do is I offered lizards a mealworm every other day. And what I try to do is match up what lizards were doing the same trials either one day after eating or the same day of it. So the day after they ate or right before they ate on that day, I tried to make sure that lizards were engaging in the same trials, the same behavioral trials, so there wasn't variation in that from individual to individual. It's not a perfect science because some lizards just wouldn't eat when they're in the lab because they're stressed, and so you just lead them to the water and hope they drink, kind of thing. So yeah. Yes.
Actually, another question. This is just because I don't know dermatology enough, but so in the first chapter with the different temperatures, depending on where you put the markers.
Mm-hmm.
Does sustenance, how full the animal is, affect its body temperature?
It can, yeah. So it can also, and so these operative models are not a perfect science, because there can also be things like you can be in the same microhabitat, oriented a little bit different, and so your angle to the sun is different, and that can influence your body temperature too. So what I try to, I obviously can't control for the food issue, but what I try to do is make sure that in my different models in the same microhabitat, I was varying the position that they were in, kind of like north, south, east, or west. Are they vertical? Are they kind of horizontal? You know, so just kind of trying to get a wide variety of those things. But there are lots of different things that can influence body temperature, and how far off you from the substrate. Are you pushing up or not? You know, so just try to account for that with variation in the models. You know. Anybody else? I don't know if I'm not on this chat. I don't know how to get there. So if anyone has asked me a question on there, I'm sorry.
Just minimize.
Okay, that's a great idea. Okay. It'd be embarrassing if no one asked any questions. Okay, chat. Oh, okay. Well, people said great job. I'll take that. Does anyone else have any questions? I had so much fun. Arizona is a lovely place with good beer and good scenery and good lizards. I was gonna say good friends, but I was by myself. So okay. Yep. Okay. Well, thanks everyone.
All right.
Can I just get off this now? Okay, thanks everyone.
Hey Tyler,
Yeah.
am I getting back on this link?
That's a good question. Yeah, let's do that.
Okay. Where is your defense?
I think we're gonna head on over pretty soon.
Okay. All right. So maybe I'll just jump back in like 10 minutes, 5 minutes, 10 minutes.
That should work. Yeah, I'll
Okay.
have someone shoot you a message maybe if we're running late.
Okay.
But that
Yeah.
Okay, thanks.
Sweet.
Hey, does anyone know if I recorded this, how to find the recording? Oh.
You like
Oh, I love it. Oh.